Imagine, for just a second, that you have the complete, pristine blueprint to human disease sitting right in front of you.
Which is, the ultimate dream in biology. I mean, that's what we all want.
Exactly. But the only catch here is that this blueprint is written in a make things infinitely more frustrating if you're looking for the actual typos that cause complex diseases, things like diabetes, autoimmune disorders, or heart disease. You usually aren't going to find them inside the genes themselves.
Right, yeah. They're hiding out in the vast, seemingly empty, dark spaces between the genes.
Which is just wild to think about. How on earth do you find the exact single-letter typo responsible for a disease when it's buried in this absolute ocean of genetic dark matter?
It is a monumental problem.
Yeah.
I mean, we used to call that space junk DNA, which is laughable now because we know those dark spaces are actually the control panels for our biology. But pinpointing the exact single-letter change that flips a disease switch in those regions, it has been probably one of the biggest bottlenecks in modern genetics.
Which brings us to the actual mission of our deep dive today. We are pulling apart a massive 2026 study published in Nature by Siraj and colleagues. It's called Functional Dissection of Complex Trait Variants at Single Nucleotide Resolution. And what this research team did is kind of mind-bending.
Absolutely.
They didn't just use computer models to guess where the typos might be. They physically built and tested over 220,000 specific genetic variants in the lab to find the exact molecular switches driving human traits.
And to really appreciate the scale of that, testing 220,000 variants, we have to look at how we normally hunt for disease genes.
Okay, let's unpack this using an analogy, because the jump from statistical guessing to physical testing is huge. In genetics, we have these massive surveys called genome-wide association studies, or GWAS.
Right, the classic GWAS.
If we're hunting for a culprit causing a disease, GWAS is like looking at a map and figuring out which specific city the criminal lives in. Then scientists use a statistical method called fine mapping. That narrows the search down from the whole city to a specific apartment building. It gives us a list of suspects, what they call a 95% credible set.
But the problem is, the search usually stalls right at the front door of that building.
It stalls because of a phenomenon called linkage disequilibrium, right?
Exactly. Linkage disequilibrium. And this is where this statistical map gets incredibly blurry. It's essentially when chunks of DNA are inherited together through generations.
Like a package deal.
Right? Think of it like a deck of cards that hasn't been shuffled thoroughly. Certain cards always end up being dealt together. In your genome, because nearby genetic variants travel together as a solid block through human history, a completely harmless variant might always sit right next to the actual disease-causing variant.
So going back to the apartment building analogy, they're roommates.
Yes. Roommates. And because they always show up together in the patient data, statistical math alone just cannot tell you which roommate actually committed the crime.
So the math hits a wall. And that is where this new paper comes in. Instead of just looking at the statistics, this study is the equivalent of kicking down the door of the apartment building, interrogating every single occupant inside, and chemically proving which one is the culprit.
It's a brute force biological approach.
But physically doing that sounds impossible. How do you mechanically interrogate 220,000 microscopic doors at once?
You build what the researchers call the MPRA engine, a massively parallel reporter assay. It was a brilliant piece of biological engineering. To test these non-coding regions, the control panels we call cis-regulatory elements, they had to take the DNA out of the human and put it into a highly controlled testing environment. First, they literally synthesized short, custom snippets of DNA from scratch.
Like printing DNA on a microscopic scale?
Yes. Millions of tiny custom strands. They made them exactly 200 base pairs long.
Why 200?
That length is crucial because it's long enough to capture the surrounding biological context, the neighborhood, but short enough to manufacture efficiently.
Wait.
In the exact center of each snippet, right at position 100, they placed either the reference allele, the normal letter found in most people, or the alternative allele, the mutated letter they suspect might cause disease.
So they have two identical 200-letter strings of DNA, but one has, say, a C in the middle and the other has a T.
Precisely.
But if you put 220,000 of these microscopic strings into a liquid solution, they're completely invisible. How do you keep track of which string is which once the experiment actually starts?
They attached a tracking device to the end of every single sequence, specifically in a region called the 3' untranslated region. They stitched on a unique 20 base pair barcode.
Almost like putting a microscopic license plate on every single genetic car they build.
That's a perfect way to visualize it. Next, they took all these barcoded snippets and cloned them into plasmids. A plasmid is a small circular piece of DNA that naturally exists in bacteria, but scientists use them as delivery vehicles.
Why use a circle instead of just putting it into normal human chromosomes?
It's purely logistical. Inserting DNA into a genome at the exact same spot 220,000 times is a nightmare. Plasmids just float into the cell and act like independent little factories.
But a piece of DNA floating in a circle won't do anything on its own, right? It needs a motor to actually turn on.
Which is why they engineered the plasmid to include a minimal promoter. Think of a promoter as a basic landing pad for the cell's reading machinery. It's an engine idling in neutral. If the 200-letter snippet they attached contains an active regulatory switch, it'll hit the gas pedal, and the cell will start transcribing that sequence into RNA.
I see where this is going. The cell reads the custom DNA, and it spits out an RNA copy. And because that RNA copy includes the unique barcode, that microscopic license plate, you just have to count the license plates at the end of the day.
Exactly. The RNA is essentially a biological receipt. They took these engineered plasmids and inserted them into 5 completely different human cell types: K562 for blood, HepG2 for liver, SKNSH for brain, HCT116 for colon, and A549 for lung.
I assume they used 5 different tissues because a genetic switch that turns on a liver function might be completely ignored by a brain cell.
Biology is highly context-dependent, exactly. So they let the cells incubate, and then they sequence the RNA. If they saw a massive spike in a specific barcode's RNA compared to the DNA they put in, they knew the specific 200 base pair snippet contained a powerful switch.
And if they compare the normal one to the mutated one?
Right. If the snippet with the normal letter produced 100 RNA receipts, but the snippet with the mutated letter produced 1,000 RNA receipts, they just proved that a single letter change alters gene expression. They call these expression modulating variants, or MVARs.
The sheer scale of this operation is just staggering to think about. They tested over 221,000 fine-mapped variants, plus another 86,000 control sequences just to make sure the assay was working. And out of all that, they isolated 13,121 high-confidence trait-associated regulatory variants, TARVs. They proved these specific single-letter typos change biological function.
It's a massive data set.
But I want to pause here and look at the actual performance metrics because there is a catch. The paper states they achieved a precision of 0.83, but a recall of only 0.20 for complex traits.
Yeah, that is the fundamental trade-off of this technology.
Right, so I want to do a quick recap here to make sure we're all on the same page before we move on. Point 1, GWS and fine mapping narrow down the genetic neighborhood, but linkage disequilibrium hides the true causal variant.
The roommates traveling together, right?
Point 2. To solve this, they built the MPRA engine using 200 base pair DNA snippets, barcodes, and a minimal promoter in 5 cell lines to measure RNA output.
And point 3. The result was 13,000 highly precise TARVs identified out of over 220,000 tested.
But getting back to those stats, if the precision is 83%, that means when this engine finds a switch, we are highly confident it's real, but a recall of 20%. That means the engine entirely missed 80% of the active switches. Why is the flashlight missing so much of the room?
It comes back to the delivery vehicle we talked about earlier, the plasmid. A plasmid is a small, naked circle of DNA. But inside your actual cells, your DNA is not a naked string. It is tightly spooled and folded into incredibly complex 3D structures called chromatin.
Like an impossibly tangled ball of yarn.
Yes. And many genetic switches only work because of how that yarn is folded. A switch might be 1,000,000 letters away from a gene, but because the DNA loops back on itself, they physically touch. An episomal plasmid completely lacks that 3D architecture. Furthermore, the assay might miss variants that only act as isolated brakes repressors because the minimal promoter is already idling so low. Or, as you pointed out earlier, maybe the switch only turns on in connective tissue, which wasn't one of the 5 cell types they tested.
So it's an incredibly precise tool, but it requires a very specific environment to work. Still, finding 13,000 guaranteed molecular switches is a huge leap forward, which makes you wonder, if you could run this custom barcode assay on your own DNA, which specific trait or health risk would you want to physically test every single letter for?
Oh, the ability to peek under the hood like that is compelling. But the study reveals that finding a single switch is just the beginning of the complexity.
Because what happens if you have 2 typos in the exact same room? This was one of the most fascinating parts of the paper for me. We naturally assume genes operate in a straight line. One mutation causes one problem. But the researchers tested variant pairs that sat within 150 base pairs of each other, testing all 4 possible combinations of those 2 spots, and they found that 11% of these pairs interact in a totally non-additive way.
They call this regulatory epistasis. It means the variants are forming secret alliances. The biological math stops behaving normally. 1 + 1 suddenly does not equal 2.
Let's ground this in one of the clinical cases they highlighted. I remember they looked at the ESS2 locus, which is tied to DiGeorge syndrome, a disorder that can cause heart defects and immune issues. They looked at 2 specific variants sitting right next to each other.
They did. And on their own, both of those individual mutations activate transcription. They each alter a docking station on the DNA called a CTCF motif, slightly turning up the volume of the gene. But when a patient has both mutations on the exact same strand of DNA, the volume doesn't just double. The combined effect is massive. It's synergistic amplification. In this case, 1 + 1 = 3.
So they act like an amplifier circuit, driving the gene into overdrive. But the paper also showed the exact opposite phenomenon. There was a locus THBS2, which is associated with systolic blood pressure, and the 2 variants they tested there were effectively silent on their own. They did absolutely nothing.
They were biologically inert until you combine them. When both of those spots happen to mutate to a C allele simultaneously, they chemically bind together to create a brand new docking station, a gin-binding motif out of thin air.
Wow, it's like 2 useless halves of a broken key. Separately, they can't open anything. But if they happen to slide together, suddenly they form a perfect key. Mass amounts of proteins rush in to bind to it, and gene expression spikes. That is wild.
But the most profound discovery regarding these interacting pairs has to do with human evolution. The researchers notice a stark pattern. Most of the interacting variants they found don't amplify each other. They actually dampen each other.
Like pressing the gas pedal and the brake at the very same time?
Precisely. They call it linkage masking. Imagine a severe disease-causing mutation arises in a population. Normally, evolution would weed that out quickly because the individual would get sick. But what if, entirely by chance, a second mutation happens right next to it that does the exact opposite?
The opposing genetic effects cancel each other out.
Yes. The harmful variant is now masked. It can safely hide in the human population, silently passing from generation to generation because its roommate variant is acting as a molecular brake.
That is both brilliant and slightly terrifying. Let's do another quick mental recap here. Point 1. Regulatory epistasis shows that 11% of nearby variants interact non-additively.
Right, they form alliances.
Point 2. We saw in the ESS2 DiGeorge case how effects can amplify, or in the THBS2 blood pressure case, how a combo can unlock a gene out of nowhere. And point 3, linkage masking. Variants often evolve to cancel each other out, hiding from natural selection.
Which is a huge paradigm shift for how we view inherited risk.
Think about your own family history for a second. Could a severe trait be hiding in your DNA right now, perfectly masked by another tiny typo acting as a brake pedal?
It is highly likely we all carry masked variants. But understanding these brakes and gas pedals require the researchers to confront a massive blind spot in our current knowledge.
Right, because I have to bring up the 31% problem. The paper points out that roughly 1/3 of these highly active disease-causing switches don't make any sense. They don't match any known transcription factor binding rules. They break the mold of how we think DNA regulates genes. If they don't match the known biological grammar, how do you even begin to translate them?
You have to force the DNA to show its work. For 136 of these mysterious rule-breaking variants, the researchers deployed a technique called saturation mutagenesis, also known as deep mutational scanning. They stopped relying on the natural typos found in human populations. Instead, they went into the lab and systematically forced every single possible letter substitution across the entire 200 base pair element.
So if the natural sequence is ATCG, they artificially built TTCG, CTCG, GTCG, and ran that process for every single one of the 200 positions.
Over 170,000 unique artificial mutations were synthesized and run through that MPRA barcode engine we talked about earlier. They mapped every conceivable consequence of altering that strip of DNA.
But testing 170,000 random mutations has to create a mountain of noisy data. The paper mentioned they used Gaussian filtering to clean it up. Wait, isn't Gaussian filtering an astronomy tool? Like how the James Webb Space Telescope processes images to see through cosmic dust?
It is the exact same mathematical principle. When an astronomer looks at a raw image of deep space, there is random background static noise from cosmic rays or sensor heat. Gaussian filtering applies a mathematical bell curve to smooth out the random spikes, allowing the true concentrated light of a distant star to shine through. Biological data has static too. Cells naturally fluctuate in how much RNA they produce. By applying this astronomy smoothing to their data, the random genetic static faded away and clear functional hotspots lit up.
The stars in the genetic dark matter.
They term these hotspots activity blocks. They're short stretches of DNA, typically only about 9 base pairs long, that are incredibly sensitive to any mutation. These 9-letter stretches are the actual functional words in the genetic sense. And the truly shocking part of mapping all these words, they discovered that 34% of these activity blocks act exclusively as repressors.
Over a third of these hidden switches exist purely to hit the brakes. If a patient gets a mutation there, they accidentally cut the brake lines. And this mapping technique led them to one of the most incredible clinical findings in the paper, the sex-dependent CDHR3 locus.
Oh, this is a perfect example of how environmental context changes the genetic rules.
Right. They tested a specific variant, a single-letter typo that regulates a gene called CDHR3. But in the assay, this typo only changed the RNA output in the HepG2 liver cells. It was completely ignored by the K562 blood cells. Now, earlier we assumed that was just because liver and blood do different jobs, but the actual reason is wild.
Saturation mutagenesis revealed that this specific typo accidentally creates a perfect docking station for 2 transcription factor proteins, SRY and SOX9. The key here is SRY. The SRY gene is located exclusively on the Y chromosome.
The male chromosome.
Yes. The HepG2 liver cells used in laboratories originally came from a male donor. They are XY cells. They produce the SRY protein. The K562 blood cells came from a female donor. They are XX cells. They do not produce the SRY protein.
So it has nothing to do with liver versus blood. It's entirely about biological sex. A woman could have this exact DNA typo, but because she lacks a Y chromosome, her body doesn't manufacture the SRY key to fit the broken lock. The typo is completely harmless. But if a man has the exact same typo, his Y chromosome produces the key, unlocks the mutated switch, and triggers the disease risk.
It is a perfectly sex-dependent genetic risk factor, explicitly proven at the molecular level.
Which completely shatters how we think about risk. Let's throw a third recap here to lock this in. Point 1. To figure out the unknown variants, they used saturation mutagenesis, testing every conceivable typo in a 200 base pair region.
170,000 of them.
Point 2. They used Gaussian filtering to find activity blocks, identifying exact 9 base pair words of DNA, and found many act as repressors.
The break lines.
And point 3. The CDHR3 case proved that a variant can only trigger if the patient is biologically male, due to Y chromosome-specific transcription factors, which leads to a massive question for anyone listening. If a specific DNA typo only triggers a disease depending on your biological sex, how does that shift our entire approach to prescribing medications? You can't just treat the symptom, you have to treat the microenvironment the DNA lives in.
It forces a total paradigm shift toward highly personalized, biologically aware medicine. But for this to be more than just a brilliant lab experiment, the researchers had to prove this translates to living, breathing humans out in the real world.
The ultimate so what test. We've talked about plasmids and liquid cultures and artificial bioengineering. How does changing one single letter in a dark non-coding region actually change the blood test results of a real person?
They chose to prove this by looking at a variant called RS111482779. Statistical fine mapping had previously shown a weak fuzzy link between this region and human HbA1c levels.
HbA1c, that's the blood test that measures your long-term blood sugar. It's the gold standard for diagnosing diabetes.
Exactly. When they ran this variant through their MPRA assay, the data showed that this specific letter change disrupts a GATA motif, meaning it breaks the docking station for a crucial protein complex that usually turns on the nearby C2 core of 88 gene.
But the critique of MPRA still stands, right? That was in a floating circle of plasmid DNA. How do we know it behaves like that in a real human chromosome?
They took the next leap, CRISPR base editing. They didn't just use CRISPR as molecular scissors to cut the DNA. They used advanced base editing to go into living, complex human cells and chemically alter just that one single letter, cleanly swapping a G for an A without breaking the DNA strand. They perfectly recreated the natural mutation. And exactly as their plasmid assay predicted, that single microscopic edit directly plummeted the expression of the C2 core of 88 gene in living cells.
That is amazing validation. But this is where this study pulls off its ultimate trick. Remember how we talked about saturation mutagenesis? How they forced all those artificial theoretical mutations just to map the rules?
The deep mutational scanning, yes.
Well, that theoretical lab data showed that if you mutated other specific letters right next to this variant, letters that don't normally mutate in the human population, you would destroy the gene's activity even more severely. It was a purely hypothetical super break created in a petri dish. But the researchers wondered, does this hypothetical superbraid exist in the real world? So they gained access to the UK Biobank, a massive database containing the fully sequenced genomes and medical records of over 400,000 actual human beings. And they ran a search for these artificial lab-generated mutations.
Out of 400,000 people, they found exactly 39 individuals walking around who naturally carried those highly severe, incredibly rare mutations.
And when they pulled the medical charts for those 39 specific strangers, their HbA1c blood sugar levels were significantly quantifiably lower than the rest of the population. Their real-world blood tests matched the theoretical petri dish prediction perfectly.
It is an absolute triumph of predictive biology, and they didn't stop there. They also used the data to identify a slightly more common version of this protector allele that appears almost exclusively in populations of African ancestry, which also significantly reduces HbA1c. They took a theoretical mechanism, validated it in a dish, proved it with CRISPR, and then predicted the physiology of real humans on the street.
Let's do our final recap before we bring it all together. Point 1. For clinical translation, variant RS191114827 directly lowers C2 core V8 expression, affecting blood sugar.
Right.
Point 2, CRISPR-based editing confirmed the MPRA lab results in living cells.
Which bridges the gap from plasmid to chromosome.
And point 3, biobank validation. Theoretical severe mutations generated in the lab were found in 39 real patients, perfectly predicting their low blood sugar levels. So, bridging all of this together, we started with the blurry, zoomed-out map of GDWAS and the statistical guesswork of fine mapping, blinded by linkage disequilibrium.
The roommates.
We used the MPRA engine with its custom barcodes to physically test 220,000 doors, finding 13,000 exact switches. We discovered these switches form secret alliances, amplifying each other or masking each other to hide from evolutionary selection. We used deep mutational scanning and astronomy algorithms to find the hidden 9-letter words of our DNA. And finally, we saw how mapping those tiny words allowed scientists to accurately predict the diabetes blood test results of strangers purely from their genetic code.
When you step back and look at the broader impact, this study is a monumental bridge. For decades, statistical genetics has been trapped in the realm of correlation, knowing a biological zip code is associated with a disease, but not knowing why. The study delivers the molecular mechanism, the causation. It proves that the dark spaces between our genes are not empty. They are teeming with a highly specific interacting grammar that we are finally learning how to read.
But understanding that grammar also leaves us with a massive blinking caution sign. We learned today that genetic variants don't operate in a vacuum. They can mask each other, silently canceling out severe diseases. As modern medicine moves rapidly toward using gene editing tools like CRISPR to go in and cure diseases by flipping these genetic switches, what happens when we fix one typo, only to accidentally unmask a hidden, silent, catastrophic variant sitting right next to it? Are we ready to aggressively edit a biological language where we're just barely starting to understand the grammar? Keep that 3 billion letter blueprint in mind and the secret alliances hiding in its dark spaces the next time you read a headline claiming the ultimate disease gene has finally been found.